Hello guys, today we will talk about directional cloning into plasmid vectors. So most plasmid vectors, they have multiple cloning sites that have recognition sequences for many different restriction enzymes. Okay, so we would like to discuss only about the main points. Sometimes it is impossible to find a suitable combination of vector, target DNA and restriction enzymes which will allow directional cloning. So there are several solutions to this problem. What are the several solutions? That we need to use synthetic linkers or adapters. So this synthetic linker or adapter can be ligated to the termini of the linearized plasmid. When the plasmid is once linearized, there can be linkage of adapters or synthetic linkers. Okay, onto the linearized plasmid or onto the DNA. Okay. The fragment of foreign DNA can be amplified by PCR primers that add the desired restriction site to one or both termini. So if this is a DNA, this is amplified by using a primer which will add the restriction site. It will add the restriction site to the both the ends of the DNA. Another is DNA fragments with recessed 3' termini can be partially filled in a controlled reaction using the Klenow fragment of E. coli DNA polymerase 1. So the 3' ends, if 3' ends are recessed, then they can be partially filled in a controlled reaction using a Klenow fragment from E. coli DNA polymerase 1. Okay, so these are the possible solutions if you do not find the suitable combination of the target DNA, plasmid as well as restriction enzyme. So when we ligate the Klenow fragment at the 3' end, this process will generate the complementary termini from restriction side. Okay, that would otherwise is incompatible. So it will generate the complementary termini from the restriction site. So partial filling of this 3' end, it will eliminate the ability of the termini on the same molecule to pair with one another. Okay, so it will not allow the same molecule, the termini on the same side of molecule to pair with one another. Okay, so that the same termini will not pair with each other. Okay, so you can see here in this picture that to the recessed 3' end, DATPs are added using the Klenow fragment of E. coli DNA polymerase 1, which will allow the addition of DATP to the recessed 3' hydroxyl group, and the synthesis will occur in a 5' to 3' direction until this recessed 3' end is filled. Okay. Again, very important thing to note that if even micromolar concentration of DATP is present, it can inhibit bacteriophage T4 DNA ligase. So ligase will be inhibited by DATP. Okay. So if DATP is used as a substrate here for partial filling, the modified DNA product, it should be purified. Okay, so modified DNA product should be purified by spun column chromatography or by 2 rounds of ethanol precipitation in presence of ammonium acetate. This will remove all the DATP from the DNA precipitation. Okay, you have to remove DATP from the reaction, otherwise ligase would be inhibited. So this is a very sensitive reaction, right? So what materials are needed for directional cloning? We'll proceed with that. So materials and the requirement will already be covered in the method. So first step is digest the vector. Okay, around 10 microgram of vector is taken. that vector has to be digested and foreign DNA has to be digested with appropriate restriction enzyme. Whichever restriction site is there in the target DNA or vector site, we will digest it with that appropriate restriction enzyme. So closed circular plasmid vectors, they are prepared for directional cloning by digestion with two restriction enzymes. Okay, which will cleave different sequences. If you use two different restriction enzymes, different sequences will be cleaved and it will generate different termini. Okay, so also try to avoid the restriction site which is located between or less than, it should be greater than 12 base pair. So this is restriction site 1, this is restriction site 2. If it is greater than 12 base pair, then this is correct. If it is less than 12 base pair, then this site has to be avoided. Okay. And if the second site is located too close to the first site, then what will happen? So you will write it in the comment box that what will happen if there is less than 12 base pair difference between the two restriction site, two restriction sites. Okay, so I'm proceeding further. You have to write it in the comment box. What will happen if the distance between the two restriction site is less than 12 base pair? Okay, think what can happen. So it should be greater than 12 base pair. Okay, so after what you will do, so you will digest with the first restriction enzyme. Okay, you will digest the target vector or the foreign DNA with the first restriction enzyme. You have to check it in the gel. After it has been cleaved, the second site will be located too close to the end of the linear DNA. So once it is digested, once, if this is site 1 and if this is site 2, okay, so if it's digested with the first enzyme, it will be linearized. Okay, it will be linearized, right? So after first is digested, it is linearized. Okay, now we will digest with the second enzyme. Okay, now another very important instruction is to determine if the two restriction enzymes work optimally in the same digestion buffer or not. Okay, if there is the same digestion buffer which can be used for both the restriction enzymes, then what we will do, we will carry out the digestion simultaneously together. We will add two enzymes together. But if the two restriction enzymes require different buffer, then it is best to carry out two digestion sequentially. And the enzyme, which now you must be thinking which restriction enzymes I should use first. So the enzymes which will prefer the lower concentration of the salt should be used first. Okay, so you have to analyze an aliquot of a DNA by gel electrophoresis, whether the first enzyme have digested it or not. To confirm whether it has been digested, it has been linearized or not. Circular band you will see towards the top, okay, and linearized towards the downside. Okay, so did I tell you right? This is the circular one which can move further. So I told just opposite. Okay, you have to check in the gel whether my band is linearized or not. Okay, you have to use, always you have to use a reference where you can compare that this band is for circular and this band is for linear. Okay, you have to check in the gel. This is the reference. The first column is the reference and this is our target study. Okay, you have to check the band is linearized or not. Okay, if it is linearized, then you can proceed with the second restriction digestion. Okay, then you can adjust the salt concentration appropriately and add the second enzyme. Okay, the next is, once you have digested, once restriction digestion have been done properly, successfully, then you have to purify the digested foreign DNA by using phenol chloroform and standard ethanol precipitation. As we have learned that purification is by phenol chloroform or standard ethanol precipitation. In my last video, in the troubleshooting video for plasmid isolation, I have discussed that how phenol chloroform and ethanol precipitation will help to remove the contamination in the DNA extract. Okay, so it is necessary to isolate the target fragments by neutral agarose or polyacrylamide gel electrophoresis. Okay, and this purification is done when there are many restriction fragments in the preparation of foreign DNA that can ligate to the vector. Okay, so many investigations are done onto that to find out, to prefer to enrich the foreign sequences of interest before ligation. So before each and every step, before proceeding to the next step, you must check each step onto the gel. Okay, so that if that has been successfully done, then proceed to the next step. Otherwise, you will not be able to troubleshoot where experiment have gone wrong. Okay, so take time, do the perfect work. Now, after the so you have to purify the digested foreign DNA. Then again, you have to also purify the vector DNA. You will purify the vector DNA by spun column chromatography followed by ethanol precipitation. Okay, so by spun column chromatography, it will eliminate from the vector preparation any small fragment of DNA generated by digesting the plasmid at two closely spaced restriction site within multiple cloning sites. So there are the chances that uh, there are different sizes of the small fragments which are prepared if the sites are located too close to each other. Okay, then there are chances that the fragments of uneven size, untarget, like what to say, there the unwanted fragments will be produced, and we need to remove all the unwanted fragments which are generated due to closely spaced restriction sites, okay, within the MCS region. Now once that is done, you have to reconstitute the precipitated DNA. Reconstitute is what? Reconstitute, you have to dissolve the precipitated DNA separately in T buffer, which has a pH of 8 at a concentration of around 100 microgram per ml. You have to calculate the concentration of the DNA assuming that one base pair has a mass of 660 Dalton. Okay, you have to confirm the concentration of the two DNAs, which is vector and the target foreign DNA, by analyzing the aliquot by agarose gel electrophoresis. Depending upon the intensity of the band, you have to check which concentration of the DNA, be it vector DNA or be it a foreign DNA, which is more intense. Okay, the more intense one is having the higher concentration. Okay, so this way you have to know. Now you have to transfer the appropriate amount of DNA to 0.5 ml microcentrifuge tubes. How you will transfer it? You will label, you have now F tubes. Okay, A and D, which contains vector, only vector. Your tube B will have only the foreign DNA. Your tube C and E, what it have? It will have vector plus insert. Okay, and your F will have super helical vector. So you have to have different types of samples to compare. Okay, otherwise you will not be able to know that what is my result, how should it look like. Okay, so the molar ratio of plasmid vector is to insert the DNA fragment. It should be 1:1 in the ligation reaction. Okay, so plasmid vector is to insert should be 1:1. Now, so to the tubes A, B and C, 3 tubes A, B and C, to the tubes A, B and C, what we need to add? We will add ligation buffer and ligase enzyme, ATP and water. But to tubes D and E, what we will add? We will add no ligase enzyme. Everything is same. We will not add any enzyme. Okay, so what's the purpose of all that? So the DNA fragments, they can be added to the tube together with the water. Okay, here it is written what? You have to add the mixture first, buffer, enzyme, ATP and water. Now you also have to add the insert and the vector. So DNA fragments can be added to the tubes together with water and then warmed to 45 degrees centigrade for 5 minutes to melt any cohesive termini which is like joining to each other, that have re-annealed during fragment preparation. So what you have to do, you have to warm the DNA at 45 degrees centigrade, then you have to chill it at 0 degrees centigrade. Okay, and then you have to add to the mixture. Understood? So before the remainder of the ligation reaction are added, so you have to first do this, then you will do step number 2. So this is step 1. You have to prepare this thing. Then you do step 2. While you are keeping it in chilling at 0 degree centigrade, then you immediately prepare this mixture and then mix step 1 uh, composition with step 2. Okay, and to achieve the maximum efficiency of ligation, you have to set up the reaction as in a smaller volume as possible. Small volume reaction you will set, because the resources are so costly. So try to preserve the resources as well. Okay, and adding ATP as a component of 10x ligation buffer, it leaves more volume for vector or foreign DNA. Okay, more volume is created for the foreign DNA. And there are some ligation buffers which automatically, while at the time of preparation, ATP is already added in them. So addition of ATP is no longer needed. You have to read the content of the ligation buffer. If ATP is already added, you do not need to add it. Okay, now once all the mixture is added, step 1 plus step 2, you have to incubate the mixture overnight for 16 degrees centigrade or for 4 hours at 20 degrees centigrade. Now you must have E. coli cells, competent E. coli cells already prepared. What you will do, you will transform competent E. coli cells with dilution of each of the ligation reaction. So this ligation reaction, once it is prepared, you will use this to transform the competent cell. So how to transform the competent cell, we will discuss later on. So transformants which are arising from ligation of vector DNA alone are either due to failure of one of both restriction endonuclease. So what we see in case of tube A, okay, in case of tube A, what we added vector plus ligation mixture. Okay, their expected number of transform colonies, there should not be any transform colonies. In case of B, we have added only insert, so there should not be any transform colonies. Again, in C, it was vector plus insert, so there should be tenfold more than tube A or D. Okay, because it has both vector plus insert, it is transform colony. Fourth one is D, which has vector, which has no enzyme. This has no enzyme. Okay, E also have no enzyme. But in case of E, see, there is colonies are observed. Expected that colonies should not be there. because enzyme is not there, but still fewer colonies are seen. Okay, and in case of F, super helical vector. Okay, which is also, we expect what? We expect that there should not be any colonies, but colonies are observed. So transformants which are arising from ligation of vector DNA alone, if they have the growth of the colonies, it is either due to failure of the restriction endonuclease to digest the DNA. Okay, there was like, if we go back, it is this when even when only vector, okay, when only vector is there, and then if you observe some colonies, you there is what went wrong? It went wrong that restriction enzyme failure was there to digest one of the restriction site was not possible. Okay, and it might be incompletely digested, or there's a chance that ligation of the vector to residual amount of small fragment of multiple cloning site. Okay, there is the ligation of the vector to the residual amount, to a very small amount of the small fragment of multiple cloning site. Very small fragment have been ligated instead of whole foreign DNA. Very small fragment has been ligated to the vector DNA. Okay, which will also produce some colonies. So that's it for directional cloning. We will discuss it further. You can put your open-ended questions in the comment box. Further, if you have any question, we can solve it. So thank you. Please like and subscribe my channel for more videos. It will help. It will encourage me to make more videos like this for you all. Thank you so much.